We will be starting with the types of culture media. Culture media is the growth media for bacteria. There are different types of media. General culture media, where the gram positive and gram negative, both the bacteria can be cultured. The nutrient agar, it is the nutrient component. The agar is the component which adds the nutritional value. And the blood agar, these agar having blood as a component, chocolate agar are the most common media for growing the human pathogens, the commonest human pathogens. The
Ma'am, couldn't understand what is agar.
agar is a media. Media means it is a food. The compositions are specially made for uh, culture of the bacterial growth. So let me complete this one. Then we can have a video representation of preparing agar and all these things. Okay. It is a jelly-like substance consisting of polysaccharides obtained from cell wall of some species of red algae. Agar is a jelly-like substance, and the main constituents is polysaccharide, which has been extracted from the cell wall of some red algae, and it is regularly used for the growth of the bacteria. Understood?
Yes, ma'am.
Now, there are another medium. Agar is the commonest medium. Other medias are there, selective and differential media. There are McConkey agar, which have the bile salt, lactose, and some dye. So here these are the components which have made them somewhat uh, getting a peculiar capacity to uh, grow specific varieties of bacteria, and it hinders the other growth bacteria. So it can be used as a selective media, and if you use this selective media, then the gram positive negative bacteria, E. coli and some enteric bacteria, they can grow in that media, but the gram positive bacteria and there are some other variations which won't grow. So if you have a mixture of bacterial colony and there are mixed populations of gram positive and gram negative bacteria. So in this selective media, if you culture, only the gram negative bacteria will grow, gram positive will not grow. So you can easily screen the bacterial population. There are selective media also. It is called the SS agar for isolation of specific variety of the bacteria like the Salmonella, Shigella, Vibrio cholerae. They are specially grown in this selective media, and the other variety of the bacteria can't grow in that media. So these sort of differential media and selective media, they help to screen or to differentiate different populations of the bacterial culture. Now, how it works? Go through this picture. It is a agar specially which gives some selection value, that in case of enteric bacteria, these are grown like this, Staphylococcus. Growth inhibited by bile salt and crystal violet. So the agar which is having some bile salt in this, the bacterial population which can't grow, it would give this sort of appearance. Means there is no such colony. Can you see some dotted colonies here? So it will be a control of your experiment, no growth. Now, in case of differential media, what is done in this media? These media differentiates between lactose positive and lactose negative bacteria. So there are many type of differences can be gained by manipulating the component of this agar. Here the lactose positive bacteria, if it grows, the pH indicator is neutral dye, is red or pink in an acidic environment, and in a basic environment, in alkaline environment, it gives yellow color. So the pink to red colonies, if you can see, then you have an idea these special type of bacteria are grown. And if the color is changed to yellow, then the lactose negative bacteria, they give this sort of change of color through the indicator, and no lactose fermentation but gives an alkaline reaction. So the lactose negative bacteria can't give the lactose fermentation reaction. So no acid is produced, and that is why the red color is not achieved. That is why you can assess, you can analyze and take the inference that this population of the bacteria which are lactose negative, they will be giving the color of the media change like this to yellow. And if there are lactose metabolism positivity, the lactic acid produced in the end product, it gives the acidic reaction, creates the environment acidic, and the color is changed to red. The indicator, the dye is given, is set in such a way, in an acidic condition it will give red color, and in alkaline condition it will give the yellow color. So these sort of media, if you use, then you can select, you can screen, you can differentiate the populations of the different bacterial colonies. The metabolic pathway. The metabolic pathway or the biochemical pathway is where a substrate will convert to a product, and that product will turn into a substrate for a different enzyme. Means the pathway, it is a cyclic pathway sometimes, where one enzyme is acting over a substrate, and that substrate will be converting to a third product, and that product will turn to another substrate by the help of any enzyme. So the reaction may be a chain-like reaction can happen. These sort of reaction may be of two types, exorganic and endorganic. Exorganic reaction where the energy is released, and endorganic reaction means where the intake of energy it is needed. So here ATP will be produced, and here ATP will be deposited, and ADP will be the byproduct. Understood? Here ADP will be added, D diphosphate will be added, triphosphate will be byproduct. So energy, the another synonym of energy is ATP. So ATP is released, and endorganic where ATP triphosphate energy is invested, and in lieu of energy the metabolic pathway is initiated, the reaction is happened, and the ADP is the byproduct, diphosphate.
Ma'am, so
How it
from
look like?
ADP to ATP, energy is released now because
Huh,
of phosphorylation. So that is why exothermic reaction type.
so what I told, just see,
Yes, ma'am,
just
that
see,
I was confirming.
huh, you just see. In this x-axis, if you plot progress of reaction, And the potential energy of my molecules are like this, means the energy, how it is released. In case of exorganic energy molecule, the reactant is there and the product is like this, the amount of energy. And in case of endorganic, you just see, it is the reactant and product is high, amount of energy input. Let's refer this one. One minute, I am just clearing your idea. Let me complete this one, then we will be giving that video. Now, the metabolic process, that is the catabolism and the anabolism. What is this? The catabolic reaction, that is why the breakdown, the generation of energy and reducing power from complex molecules. It produces small molecules for use as a waste product. So, the anabolic reaction, that is the biosynthesis, what is? It is creation and it is the breakdown. In case of anabolic biosynthesis, what happens? Construction of large molecule to serve as a cellular component. Means the biomolecules, the complex biomolecules are produced, developed. The amino acids for proteins, nucleic acid, fats and cholesterol usually consume energy. So, to produce something, you have to put energy, and to after respiration you are cleaving some larger molecules into the smaller one, and where the energy is breaking down, the catabolic reaction and the generation of the energy is having in a visible effect. Now the catabolic and anabolic pathway, if you just reconcile this one, the chemical reaction which break larger molecule into smaller one are the catabolic pathway, like the aerobic respiration, anaerobic respiration, dehydration, all are the organic molecule where with the help of some enzyme, ATP is released. So when you are just respiring, after respiration, ATP is released from an organic molecule, some byproducts are produced. You just see all the steps, it is ATP releasing. Now, what is anabolism? It is consuming in ATP because you are producing something. The organic molecule, after the investment of ATP with the help of enzyme, some other metabolites are produced, and the higher biomolecules are developed, like after protein synthesis, photosynthesis, condensation reaction, the carbohydrate, protein, all are produced. Understood? So here, when you want to develop, we want to generate some more complex biomolecule, energy or ATP is invested, and where you just want to use that biomolecule for the metabolic works, for the livelihood of a living organism, then energy is produced. There ATP is released. Here ATP is invested. In case of catabolism, ATP is released. So electron transport, what is this? The electron transport is a transport system of electron which are given, uh, which are produced, and they are giving their energy as they move through a series of electron transport reaction. So there are specific sites inside the cell where there are different electron carriers, and by as the baton in a relay race, the baton is handed over from one person to another. Here electron is passed from one cofactors from one electron carrier to another, and at that time in a chain system they move forming a electron chain and they give their energy. The oxidation reaction, reduction reaction is where molecules either gain or lose electron. Oxidation is where a molecule loses electron, and reduction is where the molecule gains electron. So the three electron transport acceptor in an electron transport chain, electron acceptors are carriers are NAD, NADP and FAD. So you can be questioned, name some electron acceptors, electron carriers. Please read all the vocabularies, the synonyms, so that you don't miss if it is coming electron acceptors and you can't recognize. Whatever electron acceptors, they are only the electron carriers or the electron transporters. So oxidation reduction reaction produces electron in a cyclic manner, and uh, electron is generated, and in another reaction the electron is lost, means invested to develop the next metabolite. Now in case of the production of the food of their own, we have already discussed about the autotrophs. Autotrophs can fix the environmental energy from the environment. Mainly they take light, photon as well as chemicals. When they take photons or light to fix the natural compounds like nitrogen, carbon, for sulfur, for production of their food of their own, we call them autotroph, and where they are just using the chemicals, they are called the chemoautotroph. Now two types of autotrophs are there, chemosynthetic and photosynthetic. Now heterotroph, already last day we have already discussed that heterotroph, hetero means other, troph means feeding. For their feeding, these populations of the bacteria, we are talking about bacteria in these sections, so I am telling them bacteria. The other organism we can talk about also like heterotroph. Now in case of bacterial heterotrophic population, these heterotrophic bacteria, they are not able to produce their food of their own. They are dependent on others. That is why hetero is coming in their name. And the fixation of the energy from the nature for their production of the food is not possible by them. Some other are fixing energy like plants, green algae, and these heterotroph directly takes the plant as their food component, or the lower ecological system animal which are taking the plant as their food. So these are called the heterotroph. In eukaryotic cell, certain biochemical processes are carried out in certain organelles like in chloroplast, photosynthesis happens, in mitochondria, respiration happens. The prokaryote lack both chloroplast and mitochondria. So many biochemical processes occur in cytoplasm. So the eukaryotes have profound mitochondria and chloroplast. So photosynthesis and respiration, there is no problem. They have specific sites. But the prokaryotes don't have these. So Cytoplasm is the only site where all the biochemical processes happen. Now, in case of cellular respiration, what does the respiration function for our body? It controls the release of chemical bond energy from large organic molecules and use the energy to sustain life. So from breaking down the large organic molecules, the energy is released and these energy help to sustain the life. Aerobic cellular respiration requires oxygen to occur. Anaerobic cellular respiration don't require oxygen to occur. It is simple, aerobic and anaerobic. Now, in case of aerobic cellular respiration, if you follow these reactions, it will be really easily understandable. In case of glucose metabolism, after respiration, what happens? A 6-carbon glucose in presence of 6 molecule of oxygen, 6 molecule carbon dioxide, 6 molecule water is produced, and the ATP or energy is the byproduct. Understood? So, in glucose, the covalent bond has chemical potential energy here only. The easiest one to get to are the CH and OH bond, and these bond breakdown of the bond happens during respiration and energy is released. It is the simplest form to understand respiration. Now, if you look at the aerobic respiration, aerobic respiration is divided into 3 processes: glycolysis, Krebs cycle and electron transport system. Just follow the glycolysis procedure. In this procedure, it is the first step and in cytoplasm it happens, and from a 6-carbon glucose, after the glycolysis in presence of oxygen, electron is produced and the 2 molecule ATP is produced and 3 carbonated pyruvic acid is the byproduct produced. This pyruvic acid is taken to the next cycle. It is called the Tca cycle or the Krebs cycle. This Krebs cycle happens in mitochondria. It is the next step of aerobic respiration, and the other byproduct, that is the electron, it is taken care of by the electron acceptor or the electron transporter or the electron carrier, that is nicotinamide adenosine hydrogen. And these NADH, it just transports that one to the electron transport system. You just see there are, in the last step it is electron transport system. So from the first step to last step, from the second step to last step, electrons which are produced as a byproduct, they are carried and they are invested in the last electron transport system. Now in the second step, the acetyl coenzyme, it is produced by the activations of the pyruvic acid, and these 3-carbon pyruvic acid is converted to 2-carbon acetyl coenzyme, and these coenzyme is the enzyme and there are substrate, and after the enzyme substrate activation, 2 molecule of ATP is produced and carbon dioxide is produced as a byproduct. These carbon dioxide is released in the nature and 2 molecules of electrons are produced in each cycle. It is a cyclic mannered reaction, and each cycle, we are talking about each cycle. And here in the last step, the electron transport system, it is well formed over the cytoplasmic area, where the electron transporter, different electron transporter molecules are present, which efficiently take up the electron and processes these electron to the next step for the further usage inside the cells. The ATP 30 2 molecule of ATP is produced in the last step. Previously 2 plus 2, 4 molecule have been produced. So at the end of a aerobic respiratory cycle, how many ATPs are produced? You may be asking the question, how many ATPs are produced in a aerobic respiration? It can come after glycolysis, then you have to write 2. After Krebs cycle, you have to write 2. And after the aerobic respiration, if it is coming, then you have to include all the 3 steps like 2 plus 2 plus 32, that is 36. Now, what is glycolysis? In a vivid way,
Ma'am, that NADP that is produced in the glycolysis, we can also break that and we can also get ATPs.
yeah, they are mainly they are acting as an acceptor. Anything can be hydrolyzed and you can produce some energy artificially, but naturally we are talking about a natural phenomenon which occurs in a cell. So naturally they are the acceptors of the electron, and when it is necessary, the NADP, NADPH, NAD means where phosphate is added, somewhere phosphate is dissociated, somewhere hydrogenation is done. All is done according to the need of the reaction. Now, if you regard how glycolysis happens, you just see we have talked about the end product of the glycolysis, that is the pyruvic acid and 2 molecule of ATPs. Now, how it is formed? You just see the 6 carbon glucose after investing ATP, because we want to break it down. Just remember, when you want to break a larger molecule in smaller subunits, then ATP is needed, and there ATP is invested. Now after the investment of ATP, this breakdown happens and the glyceraldehyde 3 phosphate is the middle product, and there also these sort of ATP is produced, 2 molecule of ATP is produced, the NAD is hydrogenated and NADH is
No,
produced
ma'am,
and
one doubt here. Ma'am, when ATP is produced, then energy is released. So if ATP is consumed, then energy should be absorbed, not released. Why it is happening like
here,
this?
no, here it is absorbing, here it is releasing, you just say ATP to ADP. So you are investing. ADP is produced, mean from try to die.
Yes, ma'am. So bond has to break. So energy has to be absorbed, not given out. Because there are three phosphate
Given
bonds.
means, given means, there is some misunderstanding. Given means we are investing this one, na? From 3 to 2. Understood?
Yes, ma'am.
Here from 2 to 3 is ATP is produced. Here ATP is produced from adenosine diphosphate to adenosine triphosphate. In this step, it is the next step. It is the first step. We are talking about the last step, na? 2 molecule of ATP is produced. Understood?
Yes, ma'am. No, ma'am. I was talking about the energy that is given out or absorbed in the system.
I am talking.
Like we have to break glucose to 3-glyceraldehyde phosphate. So energy is required. But ma'am, ATP is invested.